All right, is everybody ready to get started today? Yeah, ready to get our learn on? Fantastic. What did we talk about last class?
Brain cells.
Yeah, neurons, brain cells. What other kind of cells besides neurons? Glial cells. All right, awesome. And do glial cells do other things besides provide the structural glue that holds the good stuff together? Awesome. Cool. So were there any questions on anything that we covered last class? Anything that you might have read on your own or read in the news or whatever? Cool. Everybody feel good? Yeah? It's kind of cold out today, isn't it? Okay. All right, well, we'll try to warm things up in here. And anyway, so we're going to do this sort of back end of the anatomy discussion. We started like Drake last time at the bottom, right? The very low levels of the nervous systems at individual neuron cells. Okay, and we're going to work progressively higher up in the hierarchy, and we're going to talk about macro level structures and neuroanatomy today as well. Okay, so it's all under the umbrella of neuroanatomy, cells, but also structures. Today we're going to move up into those larger systems. Okay, remember we finished off last class talking about some of those systems. We talked about the peripheral system and the central system, right? So we had our central nervous system of the brain and the spinal cord, and of course we had spinal nerves that projected out from the spinal cord and consisted of which the peripheral nervous system consisted of. Okay, we're going to talk a lot about the central nervous system today. Okay, anybody remember what so we talked about some other systems at the end of last class. We talked about the parasympathetic nervous system and the sympathetic nervous system, right? Okay, which of those systems, or I should say, what is the neurotransmitter that is most prominently serving the parasympathetic nervous system? I'm sorry? Norepinephrine? Why do you say that? Because why? Okay, well that's not why, but it's one of the things it does, making you relax. But why is the question. Why do you remember that? Why do you know? Why do you think that's the answer? I'm sorry? Well, no, not. It's not. What do you remember it from? Oh, the video you saw. Yeah. Okay. So, anybody remember how the rap lyrics went? Do we need to watch it again? Yeah. Okay. How about we'll watch it at the end of class? Okay. Let's crush through some new material now, but when we revisit it at the end of class, see if you can pay attention to the lyrics that are also written down at the bottom of it, and it'll cue you in, and it'll help you learn. Okay, so I teach a class on learning and memory, and one of the things that we tell people also in the back end of your syllabus about some strategies for learning and memory, make things elaborate when you encode them, when you learn stuff. So what do we mean by elaborate? Put a rhythm to it. Put a rhyme to it, right? It'll help you learn, especially if this is really crazy hard stuff for you that you haven't done before. Definitely do something extra elaborative to make it fun, to make it interesting, to make it engaging. Okay? And I find those videos are one way to help do that. Okay? Did you have a question?
Yes. One of the ways, I mean, not a question, but one of the ways, the way that I remember this from a long time ago is that the EpiPens, they deliver a shot of norepinephrine to the body, and they relax your lungs, the bronchial tubes in your lungs, in order to help you breathe when you go undergo what do they call it? Anaphylactic.
Okay, so what does that make you remember, though?
That norepinephrine relaxes.
So norepinephrine relaxes. So what does that relate to?
The parasympathetic nervous system.
You sure?
Yes.
Yeah. Okay. Testing you. All right. So, anyways, we'll make sure. Okay. Like I said, make sure that you have a really good handle on some of this stuff, and some of these ways are really good ways to learn it. Okay. So, anyways, let's talk about some basic, broader aspects of what we'll call gross anatomy, not because it's disgusting, but because it's broad scale and large. Okay. So, we're we're going to cover a lot of stuff. And I've been talking pretty slow, and it's going to ramp up. So if I need to slow down at any point, if I get too fast, just let me know. All right, we're going to cover a lot of these different structures: the spinal cord, the different areas of the brain, the midbrain, the medulla, the cerebellum, the forebrain, the diencephalon, okay, and the larger aspects of the cerebral cortex that you might call the cerebrum, okay, and we'll also talk about some other areas of support systems, the ventricles, the blood systems, the arteries, and all those kind of things, okay. A lot of what we're going to cover is very closely parallel to what you'll find in other resources that are available for the class and external to the class. Sometimes I'll replicate it with different views and different figures, okay? So we're going to follow that stuff, okay? We're going to give a basic introduction to the structural neuroanatomy of the nervous system. So we've already introduced this idea that there's a central nervous system and a peripheral nervous system. Central nervous system shown here in blue and represented by everything you see on the right over here, and the peripheral system represented in yellow here, okay? Now, when we talk about the central nervous system, which is what we're going to mostly focus on today, okay, we're going to talk about this structure of the brain, okay? Now there's two, if you look at the brain, let's say this simple diagram here, one of the first things that might stick out to you is that you can divide it in half right off the bat, okay? So there's two hemispheres, or lobes, or we'll talk about lobes later, but there's two main hemispheres, a left and a right, okay? They're fairly parallel in terms of anatomical structures. They can function in sometimes different ways, but we're going to talk about the function over the next couple months of the class. Today we're going to be talking mostly about structure. Okay, so we have this basic 2' hemispheres, and we're going to systematically work them apart in terms of different structural aspects to them. Okay, and the most, what we're going to call superficial or closest to the top part of these cerebral hemispheres, is called the cerebral cortex. All right, and we'll take a closer look at the different components of that too. All right, so let's take a look at some of these cortical areas. I mentioned a moment ago that we can divide these hemispheres into lobes. Okay, so you can start at the basic level. You have one brain, then your one brain has two hemispheres. Okay, your two hemispheres each have 4 lobes. All right, there's a frontal lobe, there's a parietal lobe, a temporal lobe, and an occipital lobe. We'll talk about that in more detail as we move along. Okay, but the idea to take away from this first sort of introduction here is that sort of outermost layer of these cerebral hemispheres is what we're going to call the cerebral cortex. Okay, and it's this, what you're going to call convoluted, because it's wrapped up and bent and goes in all these crazy folds. Now, we have really specific names to characterize different aspects of these folds of our cortical tissue. All right, so when you look at something like this, you see how this sort of in the middle here is a folded up area? Now what we can do is, if you look at this at just a very basic level at first pass, these folds can be characterized in 2 main features. Basically the peaks and the valleys. Right? So you can have a thing like this, if it's folded in this way, you have the peak or the crest of the wave, and of course the trough or the bottom. All right? And they have really specific names. And so if it's a top of a fold, we call that a gyrus. Okay, so you might refer to the left inferior frontal gyrus. But at the same time, what if you're not talking about that area of the cortex where that fold is, but instead where it's folded? Okay, that's sort of the valley in between the 2 different peaks. All right, we call that a sulcus. So you might also refer to the left inferior frontal sulcus. All right, now the gyrus, or a gyrus, if you're going to refer to several of them, we call it gyri. Sort of like your syllabus. If you have multiple classes with multiple of them, we would say you have multiple syllabi. Make sense? Same thing with the sulcus. It's a sulci in the plural. So you'll see these terms interchanged through the rest of the class, and I expect that everyone will kind of be on board with how we're referring to these things. Okay? One easy way to do that, and one easy way to remember the difference between these things, is if you are down in the dumps, just having a rough day, okay? Maybe you just lost your job or something. You had, like, I don't know, Board of Regents meeting. They're like, nope, you got to go. You're out of here. You're in a rough day, right? What would you say to characterize how you might feel? You're sulking. Yes, you're down in the valleys, okay? So easy way to remember gyrus versus sulcus is is, if you're sulking, you're going to be down. Okay, and of course the gyrus is just the opposite. Whatever is left, that's got to be the top part of the fold. All right? Okay, so we have that basic introduction. We started with one brain, two hemispheres, four lobes, and they're consisted of gyri and sulci. Let's take a closer look at some of these four lobes of the cerebral cortex. Okay, now what we're looking at here is a side view of the brain. Okay, we're obviously looking from sort of the left-hand side, looking sort of this way. So if you were looking at my face, as I'm facing here, you're looking here. Now, we're only looking at it in this way, but just to note what we introduced a few moments ago, that the brain is bilateral in its structural organization. So we're looking at this on the left side, but there's a parallel one that looks the same on the right-hand side. Okay, so just, fair introduction to that. So let's talk about these four lobes. We have a frontal, a parietal, a occipital, and a temporal lobe. Okay, that doesn't even cover all the parts of the brain. Of course, we've got this area in the back called the cerebellum. We have the brain stem that connects down to the spinal cord. That's going to run down your back. Okay, but they consist of these four major areas. You have this temporal lobe area, which is basically down and ventral and sort of out to the side, more laterally. And you have an occipital area that's going to be all the way posterior to the back here. And then we're going to have this parietal area, which is kind of like back here where your bald spot might be if you're a guy and you're middle-aged, or your dad or something like that. That's going to be sort of where the parietal cortex is going to be. And then, of course, we've got that frontal part of the brain, okay? Now, these are not haphazard designations. These are not areas where we're like, if it's counted by the frontal part of the brain, we'll just call it frontal cortex. And it's all kind of a mishmash of like hodgepodge or whatever. There's actually really clear anatomical delineations of which areas consist of which lobes. Okay, so what do I mean by that? So there's some clear anatomical markers that help to divide each of the lobes. Okay, so we'll introduce a little bit of that first. All right, so these frontal lobes, of course, is going to be obviously the frontal component, but they're separated from the parietal lobe by this major sulcus that runs right down the middle. Okay, this isn't just because the artist decided to make red on one side and blue on the other. There's a really deep sulcus that runs across these lobes. Okay, it's called the central sulcus. It's one of the main division lines, between the frontal and parietal lobes. Now, there's also another really big sulcus. Okay, it's so big that they actually call it a fissure. Okay, it's called the Sylvian fissure, and that's the fissure, the sulcus, that divides the frontal and parietal lobes from this temporal lobe area that's sort of almost functionally distinct. You can sort of pull it apart. And those of you guys remember when we worked on some of the anatomy from the beginning of class, that we were able to identify how some of these areas could be sort of separated a little bit. We were able to poke around and sort of point to the hippocampus and other medial temporal lobe areas. Okay, so you have that Sylvian fissure, or the longitudinal fissure, working this direction and separating those lobes. Okay, so that's sort of our basic overview to these main areas of each of the cerebral hemispheres. Okay, the occipital lobe, as we'll talk a lot about in the middle of the semester, is heavily involved in early visual processing, helping us to see things. Okay, if you have damage there, sometimes you go blind. You just can't see depending on the type of damage. Sometimes if it's just a tiny little bit of a damage and a tiny little bit of the visual cortex, there's only one spot in your field of view that you're blind for. Okay, and we'll talk about some of these clinical deficits in really interesting situations as we get along there. Likewise, when we talk about hearing instead of vision, we're going to talk a lot about this temporal cortex. Okay, it's where the auditory specific processing areas lie, and it's areas that are involved in helping us to produce language. But so is the frontal lobe, and we'll talk a little bit about how those different lobes are involved in different aspects of language as well. Okay, so now we're sort of covered this sort of lateral view. Let's take a look at the same sort of organization, but this time looking from the medial view. Okay, so we were looking at, if I was facing this way, we were looking here, you're looking at the outside of my brain. But let's say that a buzzsaw comes and slices me right in half here and swings me open, just like that. Okay, so I'm still looking this way, but now you're looking straight into the very middle part of the inside of the brain. Okay, so that's kind of the view that we're looking at here, and we can still see that there's a delineation across these four cerebral, what do I want to say? Different lobes of the frontal cortex. Of the cerebral cortex. All right, we have the frontal lobe up here in pink. The colors have changed a little bit, you might notice, but don't sweat that. Okay, now blue is for the parietal cortex. We have the occipital back here, and we can see the inside part, or what you might call the medial part, of the temporal lobe starting to wrap around. All right, now remember the temporal lobe is going to wrap around all the way back here too. We just can't see it because we're looking at the other angle. Okay, and this gives us a better view into these other structures that we're going to talk about too. Okay, this midbrain and hindbrain area and the cerebellum. All right, so we have our 4 lobes here, and we can look at it again from a basal view, or basically looking up from the bottom. Okay, so here we're looking at this sort of midbrain and hindbrain area, the cerebellum and the temporal lobe there. If we look at it here, we can still see that midbrain and hindbrain area. Now we're looking up from the bottom. This would be like, basically if I'm hanging out like this. Okay, so we're looking at it this way. This would be the front of the brain. Up there, this would be the back. This is where the spinal cord connects. And we'll talk about what some of these different areas are and what they do as we move along. Okay, but this is just sort of a basic introduction, sort of gross anatomy, as we said. Okay, now we've already introduced this idea of the Sylvian fissure and the central sulcus. Okay, we've got these 2 major landmarks that run all the way down from the temporal and parietal lobe and temporal parietal junction, or TPJ. And we also have that central sulcus coming all the way down there, and it's separating these frontal and parietal lobes, and particularly between what we're going to call the precentral gyrus, because it's the gyrus that's just before the sulcus. Make sure you put your phones away, guys. And also, it separates the postcentral gyrus, which is the gyrus of the cortical folds that is immediately afterwards. Okay, so you have pre and post. Okay, one way that I find that easily rememberable. Okay, obviously pre means before, and post, if anybody's like a basketball or football fan, I always think of like a post-game show after a sporting event or something like that, which is always after the fact. So it's in regards to the central sulcus. So it underscores the importance of this major landmark of the central sulcus, because we go on to define other anatomical regions in reference to it. Okay? All right, so we can take a little bit closer look at some of these sulci and gyri. Okay, so when we can look at them, particularly that precentral sulcus and the the precentral gyrus and the postcentral gyrus, the couple features jump out. There's also other names that we can use for it. Okay, so particularly people have resorted to referring to some of these areas based on their functional identifications as well. So we first identified it as a structural name, essentially precentral gyrus, which is our precentral gyrus here, and our postcentral gyrus, which is purely based on structural information. Where is it relative to the central sulcus? Okay? Now, we can also define it and refer to it based on its functional organization. And so one of the things that we'll talk a lot about when we talk about motor and sensory functions later on in the semester is this idea that there's these 2 pretty delineated areas of the brain that are independently organized to serve motor and sensory processing in particular and respectively. So in particular, what we'll talk about is this motor cortex here. Okay, it can also be called the precentral gyrus. So basically this precentral gyrus is involved in our motor control. And different sections of that premotor cortex is involved in controlling different sections of our peripheral body. Okay, and likewise, the postcentral gyrus can also be referred to as the somatosensory cortex, because it's that gyrus of cortical tissue that's involved in supporting our sensory experiences, what we touch. And just like our motor cortex is divided based on where in the body it's devoted to moving certain things, there's a similar organization, we'll call a somatotopic organization in the postcentral gyrus, of where in the body that particular area of tissue is devoted to processing sensation and touch from. Make sense? Okay. So, speaking of sensation and touch, we can look at some other basic functional characterizations of some of these cortical lobes. Okay, so we mentioned somatosensory processing being in the sort of parietal area. Okay, one of the reasons why the parietal area is, of course, involved in somatosensory processing is because of that postcentral gyrus, but it's also thought to be sort of an integration center or an association cortex, some people call it, because it's associating so many of the other sensory information signals that we're getting from vision, hearing, touch, taste, all these other information processing units to tend to converge at higher levels of cognitive processing in the parietal cortex. Okay, we'll talk about that as we move in through the rest of the class. We already mentioned the idea that visual processing seems to be organized and heavily dependent upon this posterior area of the occipital lobe, so people call that the visual cortex. Again, so we're referring to some functional names now beyond the structural ones. Okay, we can also refer to auditory cortex, or areas of the temporal lobe that are involved in processing auditory information. Okay, now the temporal lobes are highly specified for processing early auditory information, particularly in the superior temporal lobe. But if you look at other areas of the temporal lobe, let's say the very anterior part of the temporal lobe, the very anterior, we'll talk about these directions in just a minute, but this very, very frontal part of the temporal lobe there, that's going to be more involved in some language processing. And when you look more towards the medial side, basically in here, that's going to be heavily involved in processing things that we remember and helping us to learn things as well. So I'm hoping that you guys are like really activating those areas right now. Okay. And the frontal cortex is involved in what we call higher level cognition, reasoning, basically decision-making processes, impulse control, what we're going to call cognitive control of attention, and it has a lot of really strong connections that go bidirectionally between areas like the parietal cortex and the medial temporal lobe as well. So there's a lot of volleys of information that are getting bounced back and forth between the frontal and parietal networks and frontal and temporal lobe networks as well. Okay? So we've talked about this, and now we're able to look at it in both views, basically this lateral view from the outside in, and also from this medial view from the inside out. Okay, so we're able to see both of those sort of views and orientations, but you can consider this essentially as being representing one of our cerebral hemispheres. Okay, now as you might imagine, both of these cerebral hemispheres tend to work together as a team. Okay, and the way that they do that is by a series of really heavy connections that connect our two hemispheres. Anybody know what it's called? Yes, the corpus callosum. Awesome. So, what do we mean by the corpus callosum? We mean these huge bundles of white matter connectivity. What's white matter, by the way? Axons. But is it just axons? Because myelinated axons, somebody said. What is myelin? A wrapping of what? I'm sorry, a wrapping of what?
It's stuff produced by ligadendrocytes, the myelin.
Stuff. Yes, it is definitely stuff produced by ligadendrocytes. I can promise you that the answer on a test will not be stuff, but that's okay. It's a good conversation point. And what do we mean by stuff, right? We mean that sort of fatty sheath, okay? It's wrapped around the axons. And what do we say that it does in sort of a functional way? Yeah, it insulates it, exactly, kind of like insulating a wire. And wires transmit electricity, and so do axons. All right, we're kind of foreshadowing a lot of our conversations for next week. And in that transmission of electricity, we like for our wires to be insulated, right? It helps for a lot of different reasons, and we want most of our axons to be insulated as well, too. And so we have this myelination, and wherever it's myelin, it tends to be colored white. It just happens to be the color of the tissue as it is. And when there's huge bundles of these myelinated axons, we can see it pretty clearly. And that's what the corpus callosum is. It's this area. So if we're looking down from a bird's eye view of the brain, okay, and you slice kind of like in a arc. So almost like an ice cream cone scooper. Okay, so we're not just chopping down up on this top right picture. We're starting up here, and we're going. So you see it's sort of the three-dimensional view we're looking down in. And what's really nice is showing us is that we have this cortical area here. You can see it kind of in this orange or pink color. And then there's all these white lines connecting across to the other hemisphere. Okay? This area there, that's the corpus callosum, right in the middle. And we can look at that in different angles. Okay? So this would be like if you're looking down at my head from about here. Okay? This would be if you're looking at my head this way, sliced here, swung open. Okay, we'll talk about these orientations in just a minute. And this one is, of course, if You're looking at me straight this way, and a buzzsaw came and went, and then opened me, and you look straight this way. Okay? And what it shows us is different ways to see that corpus callosum connecting our two hemispheres. Okay, so you have the hemisphere on this side and this side. We can see that corpus callosum running that white matter connecting the two hemispheres. It's really thick. Okay? And we can see here, look at how big that is here. It runs all the way across. Okay? And it kind of wraps around what you might think of as a C shape. Okay? And then you can see that same really thick bundle of connections there. Okay? Now, some of the earliest and most important initial insights into cognitive neuroscience came from studies of what we call split brain patients. So these are patients who had epilepsy that was intractable, which meant it's epilepsy that was not pharmacologically pharmacologically resistant, which meant that it didn't respond to medications. And one of the ways that neurosurgeons went about treating this was to open up the skull, take their knife down through, and sever the connections between the left and right hemispheres. They severed this corpus callosum. They had a corpus callosotomy. Okay? And when you did that, what it did, it prevented the two hemispheres from communicating as much. And one of the reasons this was helpful is because epilepsy seems to be a disorder of overactivity. Okay? So it was found that these hemispheres were basically communicating too much. Hence the idea of trying to reduce some of that overactivity that might be sort of over-communicated between the hemispheres. You cut that out, and we found some really interesting insights of deficits that became evident in different visual processing in particular. We'll talk about that when we get to talk about the vision lecture. Okay? So we have this big connection here, the corpus callosum. All right. Now, we mentioned different ways that this is presented. We're looking at it this way, this way, this way, and I showed you all these weird ways to look at my head as if you were chopping me apart, right? And opening up to take a snapshot into my brain. All right. So let's take a little bit of a tour of the different anatomical orientations that we will talk about for the rest of the semester. Okay. So we'll cover them today. I expect most people have already read this stuff and is familiar with it. And I came to class prepared so that this is going to be a review of what you've already introduced. And then, of course, hopefully you guys will have a chance to go back and study and work with it a little bit. And then afterwards, it's just going to be expected that when we use these terms to refer to different areas or directions of the brain, that it's just common knowledge and that we'll be working with that language. Okay? All right. So are there any questions before we start on our anatomy orientations? All right. So if we're going to take a tour through the brain, it's helpful to have a compass, right? You want to know where your directions are. North, south, east, west, right? If you guys want to go to a cool music show in Austin, you go south by southwest. It kind of helps tell you how to get there sometimes, right? At least depending on where you're coming from. If you go to south by southwest and you're in, I don't know, Arizona, you're probably not going to get to Austin if you keep driving that direction. But it's helpful to have directions and have information that's going to tell us where to look, and to make sure we get to the right spot. So when we read an article or somebody tells us about the left inferior frontal gyrus, we know what these terms mean. Now, we talked about gyri and sulci, a gyrus and a sulcus. We're going to talk about some of these anatomical terms that are used to describe things. Okay, so here's a little bit of an easier way to conceptualize what I was showing a moment ago. And instead of using a buzzsaw to slice my head one way or another, we can do it on this napkin. Okay, which makes me feel a little bit better because I can kind of remain intact. So, a couple of different ways that we can characterize ways of looking at the brain. And we'll look at them in these different ways throughout the semester. And there's really good reasons to look in these different types of slices, because sometimes you can't even see brain structures based on the type of slice you're looking at. Okay? So, what we can do is characterize our three main fields of view. So you can basically have what we call planes. So similar to like our geometry. So you have a Cartesian coordinate or a plane in geometry. We have these planes of an axis here. So we have basically coronal, horizontal, and sagittal, and that's what we call these different slices. Okay, so let's start on this side. Okay, and this is a coronal slice. This is just cutting here, swinging the door open, looking square back in my eyes. Okay, and now what you can do is and we'll notice as we go through the class at different types of topics that we talk about, you can take different slices in different areas of the brain And of course, they're going to look different. So this would be more in this middle part. It's not going to be this big if it's up here because you're not going to have as much of the temporal lobe in it. And you're going to see this part sort of start to move away if you take this coronal slice all the way in front. All right, so this kind of takes some good like spatial reasoning skills to make sure that we can mentally keep that sort of spatial orientation in our head. All right. Likewise, we can look at a sagittal view, which is really helpful towards getting taking a medial look in the brain here towards that middle part. So here, open up, straight in this way. Okay, and we can look at this horizontal plane as well. Okay, so now you're just taking a slice clear across there, and we can see the front and the back of the brain as well. Okay, so this gives us a way to find, it gives us some insight into seeing different parts of the brain that we might find particularly useful or interesting, depending on what we're talking about. Okay, any questions? Okay, so let's see here. So we can look at that in different ways. And again, this is one of the examples of a horizontal view. And it can show us different areas of the brain. So this is our anterior part, our frontal parts here, our posterior areas where some of the occipital visual cortical processing areas are. And some things that are really nicely seen here are areas of the thalamus and our basal ganglia. And we'll talk about those structures in a few minutes, but it's helpful to start thinking about these words as we start to introduce them. Okay? We can also look in that coronal slice, and it's a different way to look at some of these structures. Now note it's a little bit more difficult to see that structure we refer to as the thalamus back here. Okay? But here we see this basal ganglia as this really sort of long structure. Okay? But here, if we look in this way, we can see the basal ganglia in very different ways. Here you can see this basal ganglia, one part of it is here, and the other part of it is here. So you see very different ways to characterize and take a look at this. Make sense? Awesome. All right, so let's talk about some of this terminology. We got to get to where we want to go, and we want to use our compass. If we don't have a compass, we just need to have people tell us, go north, south, east, west. Okay? That's kind of what we have here, our compass. We have north, east, west, south. Okay? We're going to use some of these terms to help us get to where we want to get to. Okay? So we'll be talking a lot about medial, lateral, ipsilateral, contralateral, superior, inferior, and then, of course, anterior and posterior. So what do we mean when we talk about these things? Let's take a look. All right, so you've already heard me talk about some of these phrases a little bit. Sort of about the anterior frontal lobes or the medial temporal lobes. Okay, so when we talk about medial, all right, we're talking about something very close to the midline. Okay, so if you're our midline, my midline is right down here. Okay, so if you're moving towards the midline, you're moving more medial. Okay, the opposite of that is lateral. So this is the midline. Lateral means the further and further away you get from the midline, you're moving more laterally. All right. Now, anterior and posterior, similar to as I mentioned for pre and post-central gyrus. Posterior means behind or after, like a post-game show for basketball game or a football game or something like that. So we have our posterior areas of the brain, areas more towards the rear or towards the back. We can also refer to anterior parts, our anterior frontal cortex, anterior cingulate cortex. Different areas like that refer to areas that are more towards the front. Okay? Now we can also characterize things in different directions. Okay? So in particular, we can talk about things not just the front and the back, but more towards what you might think of as the top and the bottom. Okay, so there's a couple different words that can sometimes be used interchangeably. So you're talking about moving more towards the top, you're talking about moving to a more superior view or part versus a more inferior or bottom part. All right, now there's another way that you can refer to that, is that's sort of dorsal and ventral. Okay, so ventral is going to be more towards the bottom. Think of like a vent coming down by the bottom of the door in your house or something like that. And for dorsal, the way I always remember it is I always think of Ace Ventura talking about a dorsal fin for a dolphin. Okay, now where's the dorsal fin? Yeah, it's kind of on the back, it's on the top. So anytime you're talking about something more dorsolateral prefrontal cortex, you're talking about something that's more towards the top to the lateral part of the frontal cortex. So dorsolateral prefrontal cortex out here, as opposed to dorsal medial frontal cortex. All right. So we covered some of those. Now, we talked about there, of course, being two hemispheres. Okay? So sometimes what's happening in one hemisphere of the brain is actually controlling stuff on the opposite side of the body. Okay? So when we refer to situations like that, which is very, very often, we call it contralateral processing. So you have a left-hand part of the brain controlling the right side of the body, and vice versa. Right part of the brain controlling arms and legs on the left side of the body. Okay, that's contralateral processing. But of course, there can also be what we call ipsilateral processing too, which is processing this information from the same side of the body, on the same side of the brain, or same hemisphere, that is. Make sense? Any questions? Here's a really interesting part of it. We'll talk about some aspects of the spinal cord too as we move along. But one thing that's interesting to note, sort of evolutionarily, and particularly when you'll see these terms, is it can be a little tricky sometimes when referring to like dorsal and ventral, because you have two different axes of our nervous system, right? Some of these terms go back pretty long in time when most of the species studied anatomically might have been animals. Well, animals, for the most part, walk on four legs, and they tend to operate in a plane that's kind of like this. So you have, you have your animal, right? It's got its four legs, right? Yeah. So you're working on the spinal cord, and the brain here is sort of in that same direction. As we move to two bipedal forms of walking around, which means that we're not on four legs, we had that sort of shift in our axis. Okay? So instead, this became more like this. So this might be dorsal up here. This might be ventral up here. All right, so this is like human now. But if we're looking at the spine, okay, the dorsal might be more towards the back this way, and the ventral might be more towards the front. So it changes a little bit its axis and orientation. So when we talk about the spinal cord in different areas, we talked about the dorsal roots and the ventral roots. Last class, you remember that? Okay? You have to do that sort of spatial shift in your mind to make sure that we're all on the same page. Everybody good? Awesome. All right, so pop quiz. What view of the brain are we looking at here? Frontal, anterior, okay? How about if we look at it this way? Somebody say anterior? It's not anterior. Anterior was back here. This is the front. So this is like her brain looking at you from this direction. This is Siobhan Power. She's a doctor at UC Davis. She's a cool friend. So you're looking at sort of the same angle as Siobhan's head. Let's take a look at this one. What was it? I'm sorry? Yeah, an inferior or basal view, inferior, ventral. You're looking up from the bottom. This is our spinal cord there. You have some of the cerebellum down here and some of the temporal lobe coming around there and there. Okay? How about this view? Yeah, medial sagittal. Right? So you can have a sagittal view that's lateral, right? This is very close to the midline. Why do we know that? Yes, you can see the brainstem. Exactly. And we're going to talk more about what those structures are. But if you're all the way out to the side, let's say you took a sagittal view, like just clipping off the side here of me. You know, just a little bit of a haircut. Okay? It would come down here. You wouldn't see as much of the spinal cord of the brainstem. That's much more towards the medial side. Okay? All right, so we've got this idea. And again, just as a point of note, when we talk about some of the peripheral nervous system, okay, we'll see these terms used interchangeably. And particularly, we'll add a couple more, proximal and distal. Okay, so proximal means really close towards the midline or our spine. And distal means further away, sort of like medial and lateral. Okay, we already talked about that little shift in orientation between dorsal and ventral. I know I'm doing you guys big favors with all my art. Don't worry about the art too much. This stuff is better. But There's also two big terms that are very useful to sort of drill into memory as we move along. It's an afferent and an efferent. All right, they can be really confusing. Okay, so come up with some cool mnemonics, ways to remember what these terms are referring to. Okay, is basically what direction is information flowing as it's moving through the nervous system. Okay, so if it's carrying information into a certain region of interest, let's say we're talking about carrying information into the spinal cord. Okay, that particular nerve or projection is called an afferent. So aff is going in. Now, stuff comes into the spinal cord, and of course other stuff comes out. We've already talked about that basic idea last class, right? Information going through the dorsal and ventral roots, respectively. And you can have that information carried away from the spinal cord in this particular instance. We'll call that an efferent. Okay? So they look very similar words. They sound like very similar words. But we know that there's a difference between an A and an E, right? We all cleared that kindergarten hurdle. So even though they sound alike and they look alike, and it's really easy to confuse them, do your best not to try to confuse them, and try to do a couple exercises practicing when you're studying and writing out and drawing out afferents and efferents. Okay? A couple points of some tips there. All right, so we talked a little bit about gray matter and white matter already. People asked about that, I think, last class, the difference between white matter and gray matter. We talked a little bit more today about white matter being that myelinated connections between brain areas, particularly by virtue of those myelinated axons. Okay, so these are two really big ways to delineate different aspects of the nervous system as well. All right, so we have gray matter, which is basically our superficial outside part, the top layer of our cortex. What type of a slice is this, by the way? What view? Coronal, yeah. Kind of like the beer. Okay? Right, so if you're drinking a beer, you want to be standing straight up like this. Right? You don't want to be hung over on the couch, laying it down, trying to guzzle the beer sideways. It's not really doing yourself any favors, right? You want to be standing up and drinking your beer like this, if it's a Corona. Okay, so that's the view going this way. Now, we can look and see in this coronal slice really nice ways to delineate this outside superficial layer. This is where all the bodies of the neurons are, okay? Because they're cell bodies and or soma of our neurons. They're basically, they get imaged as being gray relative to the white matter, which is representing all the connections connecting different areas of the brain. Okay? So we have that gray and white matter here. We can see that again represented in this cartoon. Okay? So we can look at our gray matter out here. You can see these organized cortical layers of cells. And our white matter instead representing these myelinated axon fibers. Okay? And we'll talk a little bit more as we get in more detail about different nucleus. If we're going to talk about a nucleus, in the plural would be a nuclei. We'll talk about them like the basal ganglia or these nuclei here, which is all these densely packed cells, but not in, say, the systematic layers of organization that you might see in this gray matter in the cortex. Okay? So you'll hear things referred to as white matter and gray matter. Hopefully we're all on board in understanding what that stuff means. We'll take a brief moment here and cover in 2 slides what people spend entire dissertations and careers talking about, which is different developmental aspects of the nervous system. How do we develop from conception to birth and then beyond? How does this nervous system develop from the conception of a new life form into this exceptionally complex array of neurons, okay? And the basic thing to keep in mind is what we'll talk about as we get into some of the larger structural identifications, basically that we have a forebrain, a midbrain, and a hindbrain, okay? And so we can look at this sort of development from very early on after conception, only 25 days, and right off the bat, people are able to identify different features that can characterize this. Mainly, we see it shown in this nice cartoon, basically blue, purple, and this grayer area. We're going to call this the neural tube. It won't be too much of a part of this class, but the idea is that these different colored sort of sections will develop into these 3 main parts of the brain: the forebrain, the midbrain, and the hindbrain. Okay?
And we can see that progression over time. Okay, so as the embryo grows, we can see that this baby has development in different areas. And as it's happening, it's starting to turn, and you can start to see that axis shift like we talked about here. So here it's kind of just straight, almost like our animal ancestors, right? But as it develops over 35, 40, 50 days, we can see this initial sort of straight sort of neural tube. It starts to bend. It starts to curve up on itself. Okay? And it'll give us our overall form that we'll see at the end. Okay? And we're not going to talk too much about development in this class. We'll talk about mostly human brains and stuff like that. But we can see these different areas, forebrain, midbrain, and hindbrain gets conserved or maintained across these periods of development. Okay? And we can see that they start to fold back and then they start to expand differentially. Okay? So we're not going to talk a lot about evolutionary psychology or neuroscience in this regard for the purposes of this class, but we can see that the cortex starts to take over a large area of development. Starts really a lot of expansion of that particular area, this forebrain that we started off. Sort of a small area part of it here becomes more relatively larger, and now it starts to take up the predominance of tissue space here, becomes larger and larger. Okay? So that's kind of the idea there. Just an introduction here of different ways you can characterize it. This looks like a crazy graph, crazy table, but it's just pointing to these same type of areas. So we're sort of, if we start from this point where we were here, this baby, okay, let's say it's born and now it's grown up into a full adult, we can see that same sort of structure here. We have the hindbrain, the midbrain, and then this forebrain, and this cortical areas that really expanded. Okay, so you can characterize all these different divisions. There's really a systematic organization here. Okay, I'm not going to have you, there's not going to be like a test question of like fill in the blanks of this graph or this table, but it's helpful to think about when you're studying in terms of placing things in the right organizational context. Okay, so we talked about this, particularly this special area of the brain that humans are particularly advanced in, which is all this extra development of the cortical sheet. Okay, now what we mean by the cortical sheet. Remember we said we have all these folds, the gyri and the sulci? You can basically imagine the cortex just spread out as one sheet. Okay, now you would kind of want a lot of it because you want more brain power, right? You want a lot more neurons. You want to pack them into this tiny little area in our cranium that's finite, right? It's really tough to get your cranium to move. Okay, so we want to get as much in there as we can so it gets convoluted and packed in and folded in our gyri and sulci. And we can look at that cortical sheet in a couple different ways. Okay, so some of the features that make up our gray matter in this cortical sheets are some characteristics that we'll talk about as we move in more depth. Okay, namely that we can look at these different layers within. Okay, so we have this pyramidal cell that we introduced in our cell morphology conversation last class, and we can see those sort of pyramidal cells here in sort of layers 4 and 5. Okay, and we can see that they're projecting the different types of neurons that are characterized in very different ways, and so we can kind of number those layers. Okay, so you don't have to know those numbers necessarily right now. I just wanted to introduce this idea. Okay. And there's connections that can be made both up and down vertically between different cells, but also lateral connections across layers. Okay, so we have that here. We have those connections that can be made. Are there any questions so far? No? Okay, so let's talk about some of these divisions that we introduced in that developmental slide a moment ago. Okay, we'll talk about these ideas of the forebrain, the midbrain, and the hindbrain, and we will talk about it in the context of how you can we're actually kind of work from the cerebral hemispheres down to some of the older evolutionary structures like the midbrain and the hindbrain we'll talk about more towards the end. Okay, so one way that's really helpful to think about this, okay, is that, and we kind of just alluded to this a moment ago, but that brains are like ogres. Anybody know why? What? They have layers. Why do ogres have layers? What? They're like onions, donkey. Yeah. All right. Cool. Everyone's awake a little bit. So, all right.
What?
I thought they were like parfaits.
Well, everybody loves parfaits. Okay? You ever hear anybody ask somebody, hey, you want some parfaits? And they say, hell no? Hell no. Okay? I love parfaits. Parfaits might be the most delicious thing in the whole world. All right. Don't get me started. So, that was quoting Shrek, by the way, for any of you who didn't catch that. I'm not really that crazy. It's actually from something. Anyways, so let's talk about these different layers as we go through, okay? Let's start with the outside, these forebrain, okay? So what we talk about are these outside cortical areas. We talk about these cerebral hemispheres, the cerebrum, essentially. So we already talked about how they're interconnected across hemispheres by virtue of the corpus callosum. There's some key structures. I just want to introduce them. We'll talk a lot about them as we get into the different systems through the course of the semester. Okay? One of the big ones we introduced the name of a few moments ago, the basal ganglia. Okay? It's this ganglia, or this group of neuron cell bodies, and they're heavily involved in controlling fine motor control, some forms of non-conscious memory, in movement control, okay? And they can be divided into 3 different areas, okay? So what we're looking at here is you can see this translucent brain. You can look straight through it in this regard. We're looking more towards that middle part, okay? We can see there's these different features. You have these nuclei in the middle. We can divide it between the putamen and the globus pallidum, and it wraps around with this large tail we'll call the caudate, okay? So that's one aspect of the forebrain. We can look at that in the other coronal slice. Okay, see how we can look at the same aspect of our basal ganglia that we saw here in this sort of lateral view. We can also look at it in this coronal view, and we can see aspects of the same basal ganglia here. Okay, so here you see the caudate, the putamen. Okay, I don't see it doesn't have the globus pallidus here, but that's part of it there. Anywho, so we can see the basal ganglia, 2 different views. Okay, it just gives you an idea. Depending on where you slice it, you'll see that tail and not. We can also talk about some of these other areas related, okay, within the sort of forebrain and this limbic system structures. Okay, some of them are the amygdala, the hippocampus, what we'll call the cingulate gyrus, can be divided between the anterior cingulate, middle cingulate, posterior cingulate, as well as what we'll call the olfactory bulb. Olfactory bulb is involved in our sense of smell. Okay, you can't see it here, but well, I'll show it to you in a moment. Well, actually, let's see here. We can show the olfactory bulb is this sort of stretch there of tissue. Those of you guys who studied the cranial nerves might recognize our olfactory nerves. Okay? So you might see where that might look familiar. The amygdala is the small almond shape. Yes, sir?
About the basal ganglia question.
Yeah.
Is the substantia nigra part of the basal ganglia?
That's a really good question. So he asked if this area of the brain called the substantia nigra, is it a part of the basal ganglia? And the answer is yes and no. Okay, it's a good question. It was actually in our test last semester, and it was a point of hot contention because I had it down for a certain answer, and people were all up in arms like, no, it's that, you had a slide, and it said this, and they were totally right, and they certainly, they got credit and whatnot. But the issue at hand that he's alluding to is that the basal ganglia is a network of structures. We mentioned the caudate, the putamen, the globus pallidus. There's also some other areas that anatomists have started to add into that network because they work so closely together. The substantia nigra is one of those areas. We'll get to it in more detail as we get more specified on the lectures about movement. But until then, let's just stick with these 3 structures, the globus pallidus, the caudate, the putamen, and we can talk a little bit more about some of these other structures in the interim. Okay? So namely, the amygdala is this very medial temporal lobe area. It's a very anterior part of the medial temporal lobe. It's about an almond-sized structure. It's easier to see if we look at it in this way. Okay? This amygdala is this almond-sized blue structure, very anterior to this area that we call the hippocampus that wraps around, sort of this C shape. It's very medial of the brain. The amygdala is heavily involved in expressions of emotions and other aspects of odor processing, whereas the hippocampus and the amygdala work together for a lot of emotional memory, because the hippocampus is thought to involve a lot of memory processing. Okay? Now, we also have the cingulate gyrus. This is all part of this limbic system, which sort of means around the rim of the ventricles and the C shape. Okay? And so we have this cingulate cortex that can be divided into the posterior cingulate, the anterior cingulate, and of course this middle cingulate part. Okay? Everybody's got all this memorized perfectly, right? All right. I'll get to it. You don't need to know the septal nuclei, don't sweat that. The hypothalamus is a really important area of the brain. It's a small area right here. It's basically right ventral to the thalamus and other very sort of diencephalic areas. Okay? The thalamus, by the way, this red area, also called the diencephalon. So you have this sort of hypothalamus in there. Now the hypothalamus is involved in a lot of autonomic nervous system control, a lot of hormonal control, a lot of aspects of very primal behaviors. Okay? Things that you might think of, and very easy to remember as the 4 Fs. Very primal type of autonomic nervous system type of things. We got feeding, so involvement of regulation of when you're hungry or when you're full. All right, feeding, fight or flight. Okay, so when you need to run or when you stay in fight, very autonomic processes, and then reproduction. So those are the 4 Fs for you. And you think about it for a minute. Okay, let us know when you're done. Let us know when you got it. You good? Okay. Not clear for anyone? Okay. Awesome. So then we have some of these limbic areas. We've mentioned a little bit about the thalamus. We're going to talk a lot about the thalamus for the whole class because it serves as a relay station. So most of our sensory information that comes in before it projects to our cortical areas that we talked about processing different things like vision, audition, touch, sometimes even smell. Okay, it gets sent to the thalamus and then projected to the cortex. So by virtue of what you call thalamocortical connections. So thalamus is heavily involved as a relay station for different sensory processing. And you can divide the thalamus into a ton of different nuclei that specifically serve these different sensory processes. All right. Now, we also talked about the hypothalamus already and the 4 Fs, fighting, fleeing. Anything else? Feeding. And reproduction. That's right. Okay. So we have that as well. Okay. Now, and we can look at that in other ways, like we've already introduced. So you have our basal ganglia here. We have the thalamus, and a couple different things there. So we have that. Let's take a look at some of the midbrain structures. Okay. So now we're going down through our layers. All right. Because brains are like ogres, and ogres are like onions because they have layers, donkey. Okay. So we'll go down to the midbrain a little bit. Now we've already talked about these cortical areas, so we're coming down past the thalamus. The midbrain is this very sort of like, very small part of the brain. It's almost just a little section here, okay? But it includes a couple of really key areas, okay? One of them we'll call the tectum, okay? So the tectum is this area sort of posterior here, okay? And let's see if I have another picture of it. Well, hold on. I'll get you a picture at the end of it's in the back of these slides. It's a really good, different way of saying it. You basically have these two little bulbs on the back there. One of them is on top and one of them is on the bottom. The top one is called the superior colliculus and the bottom one is called the inferior colliculus for obvious reasons of what we talked about, those names meaning. Okay? These also serve as very important relay nuclei for visual and auditory information processing respectively. Okay? So by respectively, what I mean is that visual processing is dependent oftentimes on relays from the superior colliculus, and the inferior colliculus tends to support a lot of auditory system relay processing. Okay? So this isn't the best way to look at the midbrain there, but it's in there. It also has some of those substantia nigra areas, nucleus accumbens, and I'll get you a better picture on that towards the end if we have time. Okay? So I mentioned the substantia nigra, also part in a lot of ways of the network supporting the basal ganglia. Okay? And there's some other areas involved in the midbrain as well. Okay? The periaqueductal gray area. Okay? That's deep in that midbrain as well. Okay? That's areas that is heavily involved in processing pain perception. Okay? As well as moderating it. Okay? Pain relief through different release of opioids and things like that we'll talk about towards the end of the class. And the reticular formation are these nuclei that drop down through here. Okay? And they're basically involved in some aspects of very primal functions, sleep, consciousness, basically if you're awake, if you're not awake, things like that, okay? We're not going to have a whole lot of time to talk about sleep, but it's a really interesting topic, and if you're pumped about it, there's really good information about it in your book, And if you have questions, just swing by the office or ask questions in class, and we can talk about that stuff in more detail. Okay? All right, let's see here. So, let's see here. Oh, we need to talk about the cerebellum a little bit too. Okay? So, we talked about the cerebellum a handful of different times. Those of you guys who saw the anatomy introduction when we had the actual brains here the first day of class, a lot of you guys got to hold the cerebellum. It's this back area, this rear posterior area of the brain. All right, etymologically, the word means little brain. Okay, now, any of you guys remember some of the stuff that we talked about the cerebellum when you had it in your hands? Oh, you're killing me, Smalls. You're supposed to remember this stuff. Yes?
It's made up of particular types of neurons called Purkinje cells.
It is made up of cells that are called Purkinje cells. We talked about that last class. It's not made up of just Purkinje cells, but it is the area where Purkinje cells are most certainly predominant. They're those big spreading dendritic spines. Remember those? Okay, so we've got the Purkinje cells in the cerebellum. There are more cells in the cerebellum than in all of the rest of the brain. Isn't that crazy? And we know like relatively next to nothing about the cerebellum compared to what we know about all the other areas of the brain. Okay? So if you guys really want some like low-hanging fruit, it's also challenging fruit to go after research-wise, but if you guys want to make a name for yourself, carve out some discoveries, check out the cerebellum. A lot to learn about it. But we do know a lot of things about it nevertheless. We know it's heavily involved in motor control processing, even some aspects of memory and some other coordination and things like that. So people who have lesions to the cerebellum, they have what's called cerebellar ataxia. You know that they get arrested for sometimes? Public drunkenness. You know why that sucks? Because they're not drunk. But they have cerebellar ataxia, so they have problems with fine motor control. So they walk around like they're drunk sometimes, and they get mistaken for being drunks around town. And that always stinks because you got to keep a bail bondsman on speed dial, bail you out, all that kind of jazz. So, anyways, so we got the cerebellum. We talked about some hindbrain areas as well related to this brain stem area. So we already mentioned the midbrain. We got the pons and the medulla. Okay. So the pons is heavily involved in a lot of sensory motor nuclei. Okay, it's where some of the cranial nerves make their connections with, and the medulla is involved in making that connection between the pons and the spinal cord. Okay, so a lot of these areas are heavily involved in very basic life-sustaining functions: breathing, respiration, heart rate. Okay, when you have tumors in these midbrain and hindbrain areas of the brain, they become very dangerous, because if they start to press and invade on some of these areas, some of the cells that are so delicate and so important that if you have those cells degenerated or pressed upon or having pressure upon them, these are the cells that control our abilities to breathe and have our heartbeats and things like that. So it's very, very essential, basic, evolutionary conserved areas of the brain, very old areas of the brain. Basic life-sustaining functions. You're also going to have some cranial nerves projecting there and things like that. This is just a bottom view of the brain, so we've already talked about that a little bit. And this is another way to take a look at the pons. This is this big area here. We've got that medulla beneath it, and that medulla is making that connection into the spinal cord beneath here, okay? You can see how big the cerebellum is on the bottom, and then of course we have our pituitary gland, which is related to the hypothalamus there underneath there, controlling a lot of endocrine function. And we talked about our olfactory bulbs and our different temporal and frontal cortexes. Okay, so this is just a different way to view it. So we've got about 10 minutes left. I'm going to zip through some support systems of the brain and some anatomy related to that. Yes, for a question.
What part of the brain is affected with, say, like a football player that sustains multiple concussions?
That's a really good question. She asked what part of the brain is affected in, say, a football player who sustained multiple concussions. So I can answer that in a couple different ways. One, I can answer it as an athlete myself. I ended my college wrestling career on a series of concussions, and I played football with that as well. And then I also did my first PhD on this at Florida, actually, studying recovery of function from mild traumatic brain injury in football players. But also, you can look at this in patients who have traumatic brain injury, by virtue of being a veteran or other types of wartime injuries or just car accidents and things like that. What part of the brain is affected? To be honest, it depends on the hit. So if you're hit from different areas, it's going to affect that area or another one. It's a really tough thing to answer though, because these are very what we call mild or diffuse injuries, and oftentimes the first, the idea is that you're we'll talk about this week because it's just perfectly into where we're going. So the brain is resting and sort of floating in this fluid, cerebral spinal fluid, and the ventricles, these sort of areas that contain the cerebral spinal fluid. And so if this is our skull and this is your brain, right? It's kind of floating in there in a healthy balance. When you have a fast thing, the inertia, the brain actually is thought to bounce or hit up against that skull. Maybe it gets bruised in some capacity or another. You have that initial sort of deficit. There could be some recovery, but the idea more broadly of what people are thinking about in those types of situations. At the more neural level, we're not entirely sure. It's a very difficult thing to study what's happening at the neural level while someone's still alive and their cranium is intact. It's a very difficult thing to study experimentally. It's a lot more easily studied in rodents and things like that. It's very difficult to replicate those types of injuries at a very mild level in rodent models and things like that. So all those qualifiers and disclaimers being said, the one idea that's very prominent is that there's what we call axonal shearing. And so the actual brain areas and those white matter connections, those axons actually get sheared by virtue of the rotational force of the inertia of the injury, and that there's damage and breakage of the axons. It's a predominating model, but we have not nailed down exactly what a concussion is in that regard. If you go to a doctor and you have one, they're going to tell you the same thing that an Egyptian shaman would have told you 5,000 years ago, which is rest, try not to hit your head again. I mean, that's what they told me. Right? They give you a couple other things, you do some other stuff, this and that, but at the end of the day, we can't give a drug, we can't give a medicine to treat that particular injury because we don't know exactly what the mechanism of action is as a target to focus on treating. But it's a really cool idea. There are some great people up in Boston, actually. Bob Cantu, Chris Nowitzki are leading a lot of cutting-edge studies on that. And one of the ideas is that based on that axonal shearing, you have these cells that might have died and released and exocytosed some of their debris, and that it might lead to what people are identifying now in, say, the Will Smith movie that you might be familiar with, chronic traumatic encephalitis, which is a sort of degenerative disease of the brain that was actually characterized decades before the guy in the movie characterized it. He just found it again and talked about it some more. But we can talk more about that maybe after class. Let's jump back to where we wanted to finish off here with talking about these other support systems. We're going to talk about the meninges, the ventricles, some of the blood vessels, and the role in protecting the brain. So the stuff in and around the brain. Okay, now many of you guys are familiar with meninges. We got to hold them in our hands. You saw me pull it off a brain the first day of class, okay? So the brain itself we're used to looking at like this. On the left-hand side here are very clear gyri and sulci, but of course, the brain is actually wrapped in a very protective sheathing. We call them meninges, and there's multiple layers. So here we have those layers start to be pulled off, and on this side, it's encapsulating it. You can see these darker parts of the blood vessels wrapped within. Okay? So we can look at these the meninges, and like I said, they consist of several layers of cells. Primarily the dura, the pia, and the arachnoid. Okay? So you can look at these different layers here. So here we have basically the outermost layer of the skull. Then the outermost layer is called dura mater, Latin for tough mother. Okay? Arachnoid is the next layer, and then there's the pia layer, which is right beneath there. And then you have this subarachnoid space, right, where there's a lot of blood vessels that are going to then work their way in there. Okay? So you can look at this in different types of slices and ways. And in this arachnoid membrane, okay, is where you're going to have production of what we're going to call cerebral spinal fluid, okay, or CSF. We'll talk a lot about this fluid as we move along. Okay, so this is a picture of when I was doing some anatomy. This is a sample of what those meninges actually look like. So we can look at the cartoons of it, or we can look at it in real life. Okay, pulled off. And what we'll also note is that it actually also wraps around the spinal cord. And we also saw that with a hands-on experience the first day of class too. Okay, we can see those blood vessels wrapped up in there as well. Okay, so one clinical example of when things go wrong here is something like meningitis, which is an infection. within the meninges, and we can see that sort of reflected here where you have this inflammation of this inflamed areas of the meninges between the skull and the tissue. Okay? So one example there. We can also have meningiomas, which are tumors that can develop within the meninges as well. So that's what that might look like. We have the brain. We can peel off that meninges and identify some of the tumors that might develop there clinically. Okay? So the meninges, they're very, very helpful and protective because you don't want stuff messing with your brain. All right? If you get whacked in the head and your skull cracks open, you still want that protective barrier. So like chemicals and dirt and other stuff don't actually get to the brain. Okay, very, very, very important protective sheathing of the brain. Now we mentioned cerebral spinal fluid, and one of the areas it's produced and resides in is within the ventricles. So the ventricles are the system of fluid that helps the brain to float, like we talked about in answering your question about concussions. Remember? Okay, so this is a lateral view of it. Okay, so we can characterize them in different ways. So one way, we can see that it's sort of that C-shaped structure. Okay, it wraps around, and there's also, we've numbered these. There's basically a first, second, third, and fourth ventricle. Now, and then there's also the cerebral aqueduct that connects the fourth ventricle down here, okay, to the third one up here. Okay. Now we can have the lateral temporal horn. It wraps around to the rear occipital horn, and then it exits, goes down through the cerebral aqueduct. Now it's made in this area shown in red here, representing this called the cerebral aqueduct. Okay, and then it goes down to the spinal cord as well. Okay, so this is different ways you can image and look at the ventricles. Okay, one clinical example of when things go awry with the ventricles is something called hydrocephalus, water in the brain. It can be something that can happen during infancy, and try not to take pictures. You got it in the book and stuff like that. It's not my call, actually. The school just doesn't want pictures of stuff during that. Anyway, I have to say it. Anyways, so hydrocephalus, too much of the cerebral spinal fluid being made. Okay, what can that look like? It can be very, 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 very problematic situation because too much fluid gets made, it compresses the neural tissue, and it can be very, very dangerous. This is an example of what it might look like. Too much cerebrospinal fluid being made. Okay, you can go and find your own images on this that are available if you'd like to see some, but other ones are a little bit more aversive, so I try not to put them forward. We can see that representative of our normal ventricular system. It becomes more expanded because too much is being produced in it. One way to treat it, okay, and this is looking down from a sort of a top view or an axial view. You can see these ventricles here much more enlarged and inflamed there. All right, is you can use a shunt that comes and you basically drill a hole in it that slowly drains that fluid out. And it's a pretty effective way to treat it. So you can save a lot of those children who might have it. So we also mentioned the blood vessels that are included in these meninges. So vascularization is a very important part of the brain. It's highly vascularized. Okay, so you can see all the blood vessels of the brain shown here, and we can also see a cartoon version of that, which replicates what we introduced from the historical lecture, tying things together about the circle of Willis. Remember Anne Green? She got hung. She woke up, didn't die, asked for a beer, made Thomas Willis famous, and he characterized what we now call the circle of Willis. All right, this sort of connection of all of these initial arteries. Okay, this is another way of looking at it. Okay, so we have basically vertebral arteries. They form this basilar artery, which then gives rise to our posterior cerebral arteries as it wraps around. You can characterize them in different ways because they project to different areas. Okay, so you have the anterior cerebral, middle cerebral, and posterior cerebral. Okay, and we can look at and see what happens when there's clinical disorders here as well. Okay, so that's basically what a stroke is. You can have a stroke happen for different reasons. The blood vessels can either get blocked or they can rupture. Okay, so different things can lead to different types of strokes. Either way, what happens is you get neural tissue damage because it's no longer receiving oxygen supplies that the blood is bringing the brain. Okay, the brain loves oxygen, it needs it, or the cells will die. If those cells die, they typically show deficits of those areas. So a lot of the areas that are typically innervated or brought oxygen from these areas, blood vessels tend to be areas in support of language. So you see language and motor skills drop people with a stroke. Okay, and you can characterize that as basically ones that are characterized by blockage or ischemic, or as well rupture, which is a hemorrhage. So you can see different examples of that. I'll post these online. Last one thing I want to mention is this idea of the blood-brain barrier, okay? You have capillaries in the brain that basically form a barrier on top of our blood vessels. And the blood vessels are carrying our nutrients, our chemicals, our gases to transfer into the brain cells. But there's this protector layer around it that won't let a lot of molecules pass through it. So sometimes when you want to introduce a drug to treat something, okay, it can't cross that blood-brain barrier. So Parkinson's has deficits in dopamine production. Okay, but you can't get dopamine into the brain because dopamine's chemical formula is too big to cross the blood-brain barrier. Okay, so what you can do though is send its precursor chemical L-DOPA, which is smaller and is able to pass this blood-brain barrier. So certain chemicals and gases can pass through, other ones can't. You can see these blood vessels; the walls of them have junctions. Okay, and so that's one way that the brain also protects itself from outside toxins and chemicals. And an example to finish off with of clinical disorders that can happen of the blood-brain barrier. You don't hear about it a lot, especially here in Dallas, but you have these junctions here that are held together by pressure from our atmosphere, keeping it very tight. When you go high up to altitude, okay, it can kill you. It can even kill a man, okay, or a woman too. But you have basically clinical situations of high altitude cerebral edema, high altitude pulmonary edema. You're so high up that the atmospheric pressure is reduced, and so the gases and molecules are able to escape through those junctions that we saw were otherwise packed really tightly. Okay, so one example of that I can tell you about from personal experience was climbing Mount Kilimanjaro. It can really kill a man. Okay, I got up to about 15,000 feet. I was like, yeah, this is freaking awesome. And then I was not prepared to go much further, but of course I kept hiking up. Got up to 19,000 at the top. It was absolutely miserable because I had both of these conditions, which are terminal, high altitude cerebral edema and pulmonary edema, which meant that all these gases and chemicals and good stuff that's usually in the blood vessels was all leaking out because the atmospheric pressure was gone. It was leaking through the blood-brain barrier. You start to get this edema, the swelling in the brain and swelling of the lungs. Okay? You start to have your functions start to drop down and then you die. Okay? I was very lucky because I was just about there and they were able to save my ass, and here I am to speak of it. But about 12,000 feet down, we were able to save that. And you reset the osmotic balance through some steroidal injections that we were able to get at some hospitals in town. So happy to tell more stories about that later, but there's some clinical examples of some applied aspects of neuroanatomy. Hope you guys liked it, and we'll catch you guys next class. Hi.
Okay, so you want to talk about the anatomy of the brain, right?
Do I know what?
Anatomy of the brain, right?
Yes,
you
I-
know how it's like, are you going to be asking questions about like images, like what they are, or are you going to be asking like function or both?
A little bit of both.
Okay, but when you're doing the anatomy, are you doing it by like definition or by image?
It doesn't quite make sense. What I would say is to know both.
Okay.
Okay, and it's always a hard question to answer because the best answer I can give you is to just sneak you the test ahead of time, so you just know what the questions are, right? But of course, I can't do that, and I know that's not what you're really asking.
Yeah.
But like the best answer is to just do that,
Yeah.
just tell you the question, which makes it really hard for me to answer it in the most accurate way.
Okay.
So what I can say is it's important to know both.
Okay.
Okay.
I like how you squatted down to talk to me.
Well, it's a little bit,
It's okay.
yeah. Hey, what's up?
So the 2016 compliance training, you know about that, right?
Very sort of not, but a little bit. I guess you guys have to do something online.
Yeah, okay, so me and Rachel didn't get it, but the other two TAs got it. So I asked Dr. Huxtable about it, and she said I wasn't on some kind of list. So do you know how I can get on that list?
I don't, but we can sort it out.
Okay.
The best thing that you can do is send me an email tonight to remind me, and then I can try to track it down.
Okay. I can email her too to see what she...
Yeah, but email me for sure, because if
Okay.
not, it might slip my mind.
Okay.
But I mean, that's administrative, so we'll sort that out.
Okay, and then last thing. So Anisha sent an email about the TA sessions, right? About all the
Yeah.
times, but there's like no announcement on e-learning. So like, can we make an announcement?
Oh
Do y'all
yeah,
have
do you
to?
have access to e-learning? You should, right?
Yeah, I have.
Yeah, if you can make an announcement, you're more than welcome
Okay,
to.
how do you do that? Because I noticed in the email. But
On the left-hand side under course heading, it says like announcements, and then you go to the announcements like main thing. The top left sort of like square, it says new announcement or create announcement.
Okay.
I'm assuming that you have access to do that. If you don't and you want some, I think I gave it that access to you, but again, email me and I'll try to sort it out.
Okay.
Yeah, I will sort it. I saw that email from, I should probably take the microphone off. Huh? Yeah. I just get used to it. I don't realize it's on. Anyways, yeah, I saw that email. I just assumed that it was sent to everyone, and I just like kind of like zipped it along. But yeah, just let me know whatever I can do, and I will do that.
Right.
But usually at the end of class, there's a lot of things that I get asked, so it's really helpful to email
Yeah, I'm
afterwards.
definitely email you. Yeah. All right. Thank you, Dr. Dante.
No problem.
Bye.
Later, ladies. What's happening?
Question.
Yes.
CSF. What makes it flow around? Is it just gravity?
Yeah, I mean, gravity's going to help to bring it down, at least through the aqueduct and down to the fourth ventricle and out.
And it just continuous creation keeps the pressure up?
Yeah, probably. You know, I can give you like a probably answer. It's
Okay.
the best I can do. I mean, I can make something up and lie, but we don't want to do
Right.
that. I'll just be honest on stuff I don't know.
I mean, there's no little heart at the brain that pumps it around.
No, I mean, there's not like an actual mechanical pump that I'm aware of. Now, there might be a molecular version of that because it obviously has to be produced from something. In the,
Right.
the choroid plexus is the area that's going to be producing that.
But it always flows down then.
As far as I'm
essentially.
aware, you know, I'm not a real
So no-
expert on CSF in terms of certainly its flow. It's a really cool question. It's a cool idea. I just never thought about it in that way.
All right.
Do you know where it drains
Thanks.
into?
In which capacity? In the shunts?
Where does it
or,
exit?
Like,
Well, it goes down into basically into the spinal cord.
and then
Then just absorbed in the body and
I thought it drained into the veins. Like it'll go into the sinuses, and the sinuses drain to the veins. So
Yeah.
technically, the heart could be, in a way, like siphoning it.
It might, but again, like I don't know the answer to that exactly. I'm not going to BS you. So we can find the answer. We can sort it out. Send me an email and we can work on it and figure it out in more detail. You're not going to have to certainly be accountable. It's for it be at that level for this class. So that's usually like the important point. That's like the important part to know is that for the purposes of this class, that information is beyond the scope of what this class is designed to cover. So we don't need to know it, really. There's a lot of stuff that we don't need to know, and we definitely don't know it. Certainly I don't, but that's okay. We know the stuff that we do need to.
okay, cool, thanks.
Yeah.
Oh,
So
I actually work at Ben Hogan's concussion clinic.
I'm sorry?
I actually work at Ben Hogan's concussion clinic over in Plano.
Okay.
So I actually had a question.
Yeah.
We always prescribe out the fMRIs.
Yeah.
What does that actually do?
So a functional fMRI is
Yeah.
actually a measurement of blood flow during, instead of taking a picture of the brain, it's taking a movie of the brain you can think of. And so what it's doing is assessing different areas of the brain that are active or less active during different cognitive tasks.
Okay.
And one of the reasons it's useful is, this is what we did when I was doing my my PhD program down in, earlier on the football players, was it can show you basically if there are areas that are exhibiting over or under activity being recruited in basic cognitive tasks and stuff like that. So it can help show a little bit of insight, but to be honest, it's not a clinical tool. It's not a diagnostic tool.
Okay. Because our doctor always does it out. She always prescribes those to like littler kids when they're coming in.
Yeah, I mean, like, it's helpful because you get to see stuff.
Yeah.
And you get to learn stuff. But it's not clinical in the sense, what I mean by that, it's not, first of all, you can't diagnose a concussion based on what shows up in those scans.
Yeah.
Because you diagnose a concussion behaviorally based on the criteria of what happened when they hit. Did they see stars? Would they go unconscious?
Yeah.
Were there cognitive deficits? Those are behavioral criteria for diagnosing. So the fMRI can't and won't diagnose in that regard, but it's also not going to give you information for treatment in a clinical context either, because we don't really know what to do to treat it in entirety either. You know what I mean? But what it does really helpfully is it sheds us some insight into understanding what might be going on and what might be normally active or abnormally inactive or overactive, which helps us as scientists to be able to start to narrow down some hypotheses that we can test about models of understanding what actually might be the culprit at hand.
Okay.
So I mean, it's all one big like science project, right? I mean,
Yeah,
as a nation of
I've
scientists.
been doing those orders, so I've kind of just been wondering what I've been kind of stamping out. So, yeah. All right. Thank you.
Cool, man. And I should say also, like, sometimes concussions, you know, they're going to come, especially if it's associated with like a traumatic brain injury as opposed to like a mild one. So mild ones, you typically don't see much in a structural scan. You're starting to see stuff. I think John Hart has some stuff coming out of Center for Brain Health looking at white matter intensities. So basically, diffusion tract, DTI. That we talked about last class and some of the white matter, you can start to see differences in the measures that you can take of how thick that white matter is or different relative intensities of it. That can be associated with that you can, but for the most part, sort of the dogma that people operate with is those injuries are too subtle. They're too subtle to show up on a structural scan. But if it's a heavier injury, a more serious traumatic one, you might see a hematoma, you might see other things that could be evident. So a lot of times they send people to MRIs for those types of situations for the same reasons they always send you to an MRI now for your shoulder or your knee, which is like basically to precaution. Like, we have the ability to take a picture. Let's just make sure if we go and see that we're not missing something that we otherwise wouldn't have seen. So let's just take a look, make sure that everything's healthy and nothing else that we need to worry about. And also, particularly if people have insurance, they're like, it's an easy way to bill. To me, it's at a practical level.
Because we're kind of the only one that works with Medicaid and Medicare, and then also workman's comp, because a lot of ones want to avoid it. So.
Oh yeah.
And auto insurance.
Oh yeah, so they just send them right through, right?
No, because we'll have to, because pretty much have to call and do everything for them, and then they don't want to, because there's only two centers in Texas that really deal with it.
I see.
But,
interesting.
Yeah.
Cool, man.
Right, thank you.
Yeah, thanks.
Hi. The brain, right? There's a lot of like small structures inside, like especially underneath the cerebrum and everything, right? And it's kind of hard to see how they all fit together. So is there like a kind of like fancy 3D model that anyone has that like you can take out the hippocampus?
Yeah.
Or you can take out the amygdala, really?
Yeah, I mean, there's lots of softwares you can probably get for that. I don't have any available. You probably get a bunch of free stuff though. If you just do that. I might have some actually. Let me check. I think I have some old CD-ROM discs from probably about 10 or 15 years ago.
Because I remember like a long time ago, like this human body 3D CDs. I got one for my birthday. I was like, oh cool, you can see all this stuff.
Yeah.
But not one for like the brain in particular, like taking it apart and.
Yeah, I mean the stuff exists. I don't tend to work with it myself, so I'm not very familiar with it. But let me see what I can dig up. Maybe I can get you something.
Yeah, cool. Thanks.
Yeah, I mean you can look for yourself too. And,
yeah,
and
there might be something online.
Google around. It's pretty easy. And if you do, there's other imaging programs like FSL or SPM. They probably have something similar like that, but less of that's more about a data level than it is an instructional level.
That's true. Okay. Cool. Thanks.
Howdy.
So in the limbic system, you talked about the singulated gyrus.
Cingulate gyrus.
Cingulate gyrus. You said it was about attention. I was wondering if somehow.
Well, it wasn't just about attention. It actually was involved in a handful of different things. Sometimes integrating attention, other times not.
Because I was wondering if you could, see a difference between, if you're comparing MR scans between an ADHD patient and a regular patient, would there be any difference at all?
Not that I'm aware of for that particular measure. It's very difficult to find biological differences related to ADHD.
ADHD, yeah, that's what I was wondering.
Structurally in particular, you're probably more likely to find such deficits with EEG. So a couple good papers to look at for that. Ali Mazahiri has a paper in biological psychology or psychiatry, probably 5 or 6 years ago now. That was fairly well cited.
Ali
Mazahiri. Yeah, he's at Birmingham right now in England, but the paper was when he was at UC Davis. He's a great researcher, fantastic, world class, does great work.
Yeah, I was just wondering, just because, recently before I came to class, I was actually in a debate with my friend, and it's talking about like ADHD. Yes,
it's a hot debate. Hot debate.
It's very hot. So I mean, I'm stuck in the middle. I have no clue what to believe, what not to believe.
Well, I mean, most people are too, right? I mean, look, if the researchers knew, there wouldn't be a debate. So like, if clinicians, if doctors had some special knowledge that you don't have, we would know. Like they wouldn't be debating. They'd be like, nope, this is what it is. But that's one of the real wonders of the brain and how little we know about it. One of the reasons why it's such a great frontier for exploration right now is because it's not like the kidney or the intestine or some other system that we have really well mapped out. And we kind of know what's right and wrong with it, and you can kind of like map it all out.
It's amazing.
We haven't really mapped these really, not just neural issues, but ADHD is a cognitive issue. And you're talking about networks of billions of neurons. How do you map that? Very, very with difficulty. And then tied into clinical, people just don't have the answers. And this is kind of like what I like to default to with questions like about the concussions and everything else. Like, if people are telling you that they know definitively how to treat this or what it is, odds are, if you're in a casino, you should make a bet that they're lying to you. Because the honest researcher and the honest clinician is going to say, we don't know for sure. There's some cool ideas, there's some interesting ideas. Some are less likely or more likely than others, but, to be real, we haven't adjudicated them yet. We don't know. One of the reasons that scientifically it's so exciting.
Yeah, that's one of the most amazing parts. Like the first day of class when you told us about that we don't know so much about the brain, it really intrigued me. I was like, what? We don't know so much about the brain? I thought we knew everything, but I was definitely under the wrong impression. So
Dude,
I was like, what?
I could spend all class here talking about all the stuff I don't know.
That's insane.
What?
That really spiked my interest like crazy.
Yeah, to me, that's the really exciting, engaging part of the field.
Yeah, you don't know. That's
who wants to go talk about old hat and all the stuff we already know? Like, how about all the stuff we don't know? And to be honest, I tell people this too. Not for every single topic we talk about, but for a lot of the topics that we talk about, a lot of the things that we'll talk about, there's somebody somewhere who disputes it or says, I don't know about that. It doesn't make for a good class because people don't feel like they're getting value all the time if they're being told that that we don't really know anything. So we don't always talk about that higher level of science in the introduction classes. We're like, no, this is the frontal cortex. It does this. Well, really, because there's people who have been working on the frontal cortex for 30, 40 years who still have a lot of debate between their models and theories about what exactly it's operating. Now, there's a ton of agreement about things, too. Right? So there's a lot of areas and different things that there is a lot of agreement upon. Well, we think it does this, and it seems like it operates this way, but we still don't really know the full mechanism and model of how it's fully working. Again, that's the exciting part, right?
So, yeah.
And
it's amazing, to be honest, first day.
It's really humbling, actually, when you start thinking about it. You're like, oh, geez, we know, a sliver and a snapshot.
Yeah.
And so much of what's published now, the new cutting edge findings, so much of what we publish is so close to basically replicating so much of what we've already known at times. So there, researchers talk in conferences or chat amongst themselves. It's still really difficult to find original or publications that are really reporting original things that we didn't know before. It happens. It comes out and they're exciting and really wow. But a lot of this stuff is so incremental, and which is helpful. We need things to move incrementally, but so much of it still kind of circles back to
something that's unique previously.
In some ways, just depends on the lens that you look at it though with.
Yeah, just out of curiosity. Just asking that question.
Cool, man.
Thanks, I appreciate it.
Yeah, no problem. Hey guys.
Hi.
Howdy.
So I just wanted to let you know, and I know it's a really big class, so this is kind of a moot point, and I won't keep your time very long. Sorry, my brain is done today.
Huh? You think yours is?
I can only imagine.
You got the chill. You got the seat to sit in. I had to talk the whole time.
I know, and you acted things out. You showed us your art degree.
Oh yeah, well you can see it's not a very prominent degree.
It's a good thing you went this direction instead.
Yeah, I had to make some life choices.
But I won't be here next week.
Okay.
And I know that doesn't make a difference to you, but now I've looked you in the face and I've told you, so I can check it off in my little box. But I'll be back the week after that. And
okay.
And
is everything okay?
Yes.
Okay.
It's for the best reason possible. We received a free vacation that I get to plan.
Yeah. That is the best reason possible.
So I'm going. But,
totally.
It's inconvenient, but it's free.
Where are you going?
The Dominican Republic.
Ah, the DR, yeah.
So it's going to be wonderful.
Do you dance bachata?
I'm sorry?
Do you dance bachata?
No, I don't.
You should.
Do I need to?
They do it very well down there.
Okay. Well,
it's one of the areas it was actually developed. Do you like Latin dancing?
I like watching them do it. I think they have an extra hip bone or something. There's no anatomist that'll confirm that, but I think it's true.
Yeah, although a lot of it comes from your knees and chest.
Well, that's a good secret to know.
It is kind of. I was told that once, but anyways, yeah, Bachacha's a really good dance they do down there.
Fantastic. Well, thank you. Anyway, I'll be back next week, and I assume you'll post your slides, and so I'll probably be reading them and stuff from there. But yay, thank you.
You'll be okay. Just keep up with the reading and stuff like that.
Okay. Thank you, sir.
Yeah, the class is actually kind of designed to facilitate that, because with so many people, like, I know things come up, people, whatever, people have to come and go and do things, and I wanted to make it so that if you need to do things like that you're not totally screwed over.
I appreciate
and
that.
You can, if you're a responsible student, and like if you know how to work independently and autonomously and like kind of take responsibility for your learning and studying, you can capture and stay right on track if you need to by reading and staying up on the things and using the workbook and the study guide. I mean, it's designed so that you can parallel that if you need to.
Perfect. Well, it's not as interesting as your lectures, but at least it covers the same material.
All right, thanks.
Yes, sir. Thank
Yeah.
you. It was nice talking to you.
Nice talking to you.
Howdy.
Hi. I have a question.
Yeah.
Kind of on the spinal cord.
Yeah.
And because there's the afferent and there's efferent neurons, and then there's also interneurons.
Yeah.
Interneurons are they just associated with spinal reflex arcs, or do they have like
Interneurons are everywhere in the brain and the spinal cord. They're associated with lots of stuff. They relay neurons for whatever the information is that is being processed.
Gotcha.
So it's not just reflexes. It can be all sorts of stuff.
That's really interesting stuff too. Like
Yeah, they're kind of mechanistic in that way. Like they kind of just do what they're told, and they might be told to do. The reasons that they might be being told to do things could be very different reasons.
Yeah.
So a spinal reflex might be one of those reasons, but there could be others.
That's really interesting. We kind of covered like in like my previous degree plan, we kind of covered a little bit on the neurological side, just like before clinicals and stuff. But it was really interesting to see like how all that worked, and
Yeah, it's cool, isn't it?
yeah, I really do love this. It's really interesting.
Oh, I'm glad you like it.
Actually, one of the people that we're working with right now is like highly they've been bed bound for a long time, so like
I see.
on their own accord. But so it's to me it's really interesting on how you lose your center of gravity and like lose your awareness of where your center of gravity should be whenever.
That is interesting. I might have a patient that comes in and talks about that later on in terms of the vestibular system as we move along in our relationship to gravity.
Yeah.
There's a lady I know, there's a very impressive story about how she worked through a tumor that was infringing on her vestibular system and ended up having vestibular surgery about 18 months ago. And she's now just starting to recover, and she's actually trying to make a candidacy for American Ninja Warrior TV show. So she's made a really impressive recovery from that vestibular type of surgery in relation to our detection of where we are in gravity.
I really like that.
I'll see what I can do. I'm trying to get her to come in and maybe talk to the class.
Do you have like a lot of people that are going to be coming in? For...
No. We've got a chock full schedule as it is, so
Yeah.
when we can fit them in, we will. But
It's really
other
good to
times,
see like experiences like from a patient view, just kind of get like a really good feel for it.
Yep.
See how. Because like vestibular wise, in one of my clinical rotations, we also worked with a couple of patients that had BPPV, and kind of like some techniques to kind of help combat the effect of it. I thought that was really cool. That was another reason why I started thinking more neurological.
Great.
Okay, sorry. Brain.
Have a good night.
You too.